Hey, good morning, everyone. It is my pleasure to introduce today's speaker, Dr. Richard Roth. Dr. Roth was born in Germany and began his academic journey with a bachelor's degree in biochemistry from the Technical University of Munich. He went on to earn his PhD in neuroscience here at Johns Hopkins University, where he conducted his doctoral research under the mentorship of Dr. Rick Hugener. His work here focused on using in vivo imaging to investigate synaptic plasticity during motor learning. Currently, Dr. Roth is a postdoctoral researcher in Dr. Jun Ding's lab at Stanford University, where he's studying how different brain circuits involved in motor learning interact to produce skilled movements. We're excited to have him join us today. Dr. Roth, please go ahead.
Great, yeah. Thank you so much for this kind introduction, Amin. And I would like to thank Reza for inviting me to present my work to you today. And yeah, it's really an honor to give the seminar to Hopkins and see so many familiar faces on the Zoom screen today. Too bad I cannot visit in person. So yeah, like Amin mentioned, I am currently a postdoc in Jun Ding's lab at Stanford University. And today I'm really excited to tell you about our recent story looking at thalamic integration of basal ganglia and cerebellar circuits during motor learning. And most of the data that I'll show you today is actually part of our recent bioRxiv preprint. If you want to find it, you can scan this code here. So I've always been fascinated by our brain's ability to learn and produce these complex and yet precise movements that we need to get through our everyday lives. And especially I realized how much learning is involved in performing even the simplest motor tasks. When my daughter first started to eat food using her hands. So at first she would try to reach for these little food pellets or puffs, I think they're called, and would not really properly grab them and not be able to retrieve them and grab them, move them to her mouth. But with several weeks of training, she would become an expert at this task and successfully grab and retrieve the food pellet. So now motor learning, of course, is not something that only happens during development, but happens throughout our lifetime. And even in older ages, we can learn and practice new skills, such as learn a new sport or play a new instrument. And for me, my main interest has been to understand how our brain controls these movements and how our brain adapts to form long-lasting memories. And motor learning, of course, is a process that is not unique to humans, but is shared across the animal kingdom, where especially the ability to use hands to handle items and food is highly conserved across humans, monkeys, and rodents. And like many of you, we are using the mouse model as a model organism, which might not be as capable as humans or monkeys, but are still able to learn a variety of motor skills, especially forelimb skills. And so now in the brain, there are several key motor circuits that are involved and take part in controlling and learning different movements. So just to highlight a couple of these motor circuits include the motor cortex and the basal ganglia with its input region, the striatum, and its output region, the SNR, or the substantia nigra pars reticulata, as well as the cerebellum, which of course you guys are all familiar with its output region, the deep cerebellar nuclei, the cerebellar nuclei. And there is also the thalamus in the middle of the brain, which is thought to act as a hub relaying a lot of the information that passes through motor circuits. That's the part of the brain that I will talk a little bit more about today. So now over the past decades, the field has made tremendous progress in understanding the role each of these brain regions play in motor control, and we're beginning to understand how they adapt during motor learning. Yet one big important question that still remains in the field is how these distinct brain motor areas interact with each other to create a single motor output and promote motor learning. And a particularly interesting question is how these two prominent and kind of parallel motor circuits, which are the motor cortical basal ganglia thalamo cortical loop, which has been a focus on the Ding lab's work and our lab's work, and the cerebellar thalamocortical loop, which of course you guys know much more about than I do, how these two circuits interact with each other to produce a cohesive motor output. And the traditional view has been that any interactions between the basal ganglia and the cerebellum would only occur on the cortical level. But intriguingly, both the basal ganglia as well as the cerebellar outputs project as a first step to the thalamus, making the thalamus a potential place of subcortical interactions between these big motor circuits. Yet the prevailing understanding is that within the thalamus, the basal ganglia and the cerebellar projection largely target segregated, separate thalamic nuclei with little interaction. However, this kind of idea that any interaction between these big systems only happen on a cortex has been already challenged from past studies that suggest subcortical interactions between the basal ganglia and the cerebellum. And one important evidence for this came from Peter Strick's group at Pittsburgh, where they discovered disynaptic connections between the cerebellum and the basal ganglia through the thalamus and back from the basal ganglia through the pons to the cerebellum. So this supports the idea that these motor systems might already interact with each other before actually reaching the cortex. And more specifically in the thalamus, early physiological studies have shown that these inhibitory output projections from the basal ganglia from the SNR are able to modulate cerebellar evoked activity in the thalamus. So for example, when this group pharmacologically activated SNR, so increasing SNR activity, they were able to block activity in the thalamus that was evoked by cerebellar stimulation, or vice versa. When they silenced SNR, they could increase thalamic activity following cerebellar activation. So when I joined Jun's lab, we really wanted to ask the question and understand whether we can identify the cellular and circuit basis of these basal ganglia and cerebellar interactions in the thalamus, and what the functional role of such interactions in the thalamus might be for motor control and motor learning. So the first thing that I did when looking at the circuit was just, okay, where do the axons from the basal ganglia outputs and the cerebellar outputs actually go to in the thalamus? So I just injected a virus expressing the red protein tomato, in this case in the SNR, so the basal ganglia output, and a virus expressing a green fluorescent protein GFP in the deep cerebellar nuclei as the cerebellar output, and then sliced the brain and looked in the thalamus to see where the axonal projection targets go. And first of all, we can see that this is already very limited to a specific part of the thalamus, so this is the ventral thalamus, with its canonical kind of basal ganglia recipient zone, the VM, ventromedial thalamus, and somewhat canonical cerebellar recipient zone, the VAL, that's how the Allen Brain Atlas calls it, the ventrointernolateral complex of the thalamus. And so here in green are the cerebellar axons, in magenta and red are the SNR axons, and they do largely target the same area, but are clearly also distinct from each other. And the area of overlap is definitely there. So you can see wide areas here, but it depends on a little bit on what section exactly you're looking at. And particularly kind of this joint area, the border area between the two regions seems to be where the largest overlap is. But of course, just the fact that there is axonal overlap at this border region doesn't really tell us whether there are individual thalamic neurons that also receive synaptic input from both the basal ganglia and the cerebellar outputs. So to test whether there are actually individual neurons that receive these inputs, I collaborated with a fellow postdoc in our lab, Fujin Huang, to perform ex vivo slice acute recording experiments, those are patch clamp experiments, to record from thalamic neurons, while at the same time optogenetically stimulating both the SNR projections to the thalamus as well as the cerebellar projections to the thalamus using a two-color optogenetic chernodopsin approach. So regular chernodopsin in the cerebellar outputs, or crimson, which is redshifted opsin in the SNR. Of course, we've also switched things around to balance this approach. And by doing that, we were indeed able to record from thalamic neurons that have a strong inhibitory input from SNR projections, which was sensitive to picrotoxin blocking, as well as on the same neuron, we had strong excitatory inputs from the deep cerebellar nuclei. And this was blocked by receptor blockers MBQX. So when we quantify these neurons that we've recorded from, roughly 30% of the neurons in the ventral thalamus, which was the region that we targeted, showed had these neurons that received dual synaptic input from both the basal ganglia output as well as the cerebellar output. Roughly another third had only SNR input, while roughly 10% had only cerebellar input. So with this limited number of cells that we recorded from, we could already kind of plot where they are located within the thalamus. And it just so turned out that most of our recordings were in the ventromedial thalamus. And there it does appear that the green cells, which are the cells with both inputs, are fairly intermingled all over. But here we've only recorded from 33 cells, because of course PatchClamp is not a very high throughput approach. And to get a better understanding of where in the thalamus these neurons that receive both inputs actually lie, we turn to a more unbiased and high throughput approach using a intersectional anterograde transsynaptic viral tracing method, taking advantage of this transsynaptic jumping property of AAV1 viruses expressing different recombinases. So in this case, we have a AAV1 expressing free in the SNR and an AAV1 expressing flippase in the deep cerebellar nuclei. So these viruses will jump one synapse downstream into the thalamus, where we have a reporter virus that is free and flip dependent. So only if a neuron in the thalamus receives both free and flip through this transsynaptic AAV1, it will turn on this YFP reporter virus. So with that, we were indeed able to find a lot of neurons in the thalamus that receive dual input and turn on this green or yellow reporter virus. And as a control in mice that we've injected with either only CRE or only FLIP, we did not see any reporter expression, which is just quantified here. We have this approach to specifically label neurons that receive both inputs from basal ganglia and cerebellar outputs. So we wanted to test where exactly in the thalamus they are. And for that, we collaborated with Tianyi Mouse Lab at the Volume Institute, who is an expert in aligning fluorescent slice images to published atlases, such as the Allen Brain Atlas or also the Praxinos Atlas. So in this case, we cut slices from the brain, so we're injected with this transsynaptic intersectional approach. They helped us register our serial sections to the Allen Brain Atlas, and that way we could now locate exactly where these EYFP reporter expressing neurons lie within the thalamus, and particularly what thalamic nuclei they are in. So now showing a kind of summary density of these labeled neurons from several mice, we could see that this density of these labeled neurons that receive both inputs is really located here in kind of the center part of the thalamus, where when we zoom in, it's really at this border region between the ventromedial thalamus and the ventroanterolateral thalamus. So suggesting that these cells that receive both inputs are kind of at the borderline where the canonical basal ganglia recipient zone and the canonical cerebellar recipient zone of the thalamus is. And so just to show a quantification of this, roughly 70% of the neurons that we labeled were in the thalamus. So there was also a large, or decent proportion of neurons that was in the zona incerta, which is just below the thalamus, which might be interesting for a different study. And within the thalamus, particularly the VM and the VAL, so the two big motor thalamic regions, show the highest percentage of these cells that receive both inputs. And I just want to mention, so this is registering or slices to the Allen brain atlas, where kind of VAL is combining VA and VL, but when we align this to the Paxenas atlas, where VA and VL is separated, we also see these cells, particularly in VL, which is the canonical main target of cerebellar outputs. So now that we have shown that these neurons in the thalamus exist that receive both inputs from basal ganglia and cerebellar projections and where they are in the thalamus, we wanted to test what their functional role might be and whether they integrate cerebellar and basal ganglia activity. So for that, we performed a multi-site calcium fiber photometry experiment where we injected GCAMP as well as implanted optic fibers, both in the SNR and the deep cerebellar nuclei, as well as in the thalamus. And we did this by combining the GCAMP expression together with this AAV1 transsynaptic approach to specifically label neurons in the thalamus that receive inputs from the SNR and the deep cerebellar nuclei. And just one further thing we did is instead of using regular GCAMP, we used a SOMA targeted GCAMP in the SNR and the deep cerebellar nuclei, so that we don't have any axonal contamination in the signal that we're recording from in the thalamus. And the signals that we're recording from the thalamus are indeed just from the cell bodies or from the neurons that receive both inputs from the SNR and the deep cerebellar nuclei. So how do these traces now look? So this is just an example stretch of activity across a little bit over a minute, where you have some activity in the SNR, activity in the deep cerebellar nuclei and the thalamus. And just by looking at it, it already looks like that the thalamus seems to reflect what is happening at its upstream regions in the SNR and the deep cerebellar nuclei. So to formally test it, we calculated the predicted thalamus signal using multiple linear regression based on the SNR and the deep cerebellar nuclei signal. And this little magenta line here is this predicted signal that's purely based on SNR and cerebellar information. And as you can see, this predicted signal lines up quite nicely with the actual observed thalamus signal. So to test how well this regression actually fits, we calculated our square values for the calculated regression and compared that to a control condition where we shuffled the SNR and DCN signals in time, so kind of offset them to create a asynchronous condition. And indeed, in the shuffle condition, the regression fit is significantly worse than in the actual observed situation. So this suggests that kind of an awake behaving mouse, the thalamus signal does reflect what is happening at its input regions in the SNR and the deep cerebellar nuclei. So now, what is actually happening during behavior, or particularly during motor learning? So for that, we trained animals on two different behaviors. First, we trained them on a head-fixed lever pushing task, where head-fixed animals are trained to push a lever or a joystick forward to receive a water reward. And mice can learn this task across the course of around 2 weeks or 10 days, 10 training sessions. And following this task, we train animals on a head-fixed locomotion behavior, where mice are just let run on a cylindrical wheel. And again, mice learn to run more, have more running bouts on this wheel over the course of 5 to 10 training sessions. And we chose these two behaviors because they're kind of different in their nature. One is only involving forelimb push, which has been shown to be involving the cortical basal ganglia circuits a lot. And the other one is a more whole body coordination, where they're learning to run on this in this head fixed situation on this cylindrical wheel. And when we now first look at the activity that we see in the SNR and the cerebellum during the head fixed lever pushing task, we can see that there is SNR activity both at the beginning of learning, so in early training stages, as well as in late training stages. But there's clearly an amplification of the signal in the SNR, particularly this later activity that seems to coincide with reward delivery. When the mice get the water reward is increased once the mice have learned to perform this task. But interestingly, in the cerebellum, we barely see any activity. There's like weak activity during the early stages of doing this lever pushing task, but less so later, and overall it's very small. And I guess just to mention this here is, for all the tracing experiments, we have been trying to target all three cerebellar nuclei kind of as a whole. Like, we have not been trying to segregate the individual contribution of individual nuclei. And for the photometry recordings, our optic fiber has been targeted mostly over the dentate nucleus, which through some retrograde and anterograde tracing seems to be very reliably projecting to the thalamic regions that we're looking at. And we next put these mice on head fixed locomotion, on this head fixed locomotion task, where again the SNR showed a strong activity at the beginning of the motion onset, which is then sustained while the mouse is running. And similarly, now the cerebellum has this strong increase in activity that lasts throughout the movement of the mouse. But interestingly, there's also no further change of this activity with the learning. So basically, both at early and late stages of this wheel running task, the SNR and the cerebellar activity is similar. So when we now look at the activity of these thalamic neurons that receive input from the basal ganglia and the cerebellum, we again see this nice activity in the thalamus that occurs while the mouse is doing this lever pushing task, which particularly shows up during later stages of learning, where both this initial peak of activity that is related to the push itself, as well as this activity that happens later and likely is driven by reward, is higher in expert animals compared to early state mice. Now, interestingly, when we look at the thalamic activity in this locomotion task, we see a strong activity that occurs in mice during the early phases of this training, which then gets weakened in mice that have been running on this task for several days. So this suggests that the activity in the thalamus, while overall is following its inputs in the SNR and the cerebellum, the adaptation of the activity in the thalamus can be distinct from the adaptation that happens at its input level in the input levels in the SNR and the deep cerebellar nuclei. So suggesting that these cells are performing some active computation before the signal is transmitted further to the cortex. And so now this is just looking at the activity, but to test whether the input of the SNR and cerebellar projection to the thalamus and these neurons in the thalamus itself actually play an important role for motor behavior and motor learning. We wanted to silence these projections as well as the neurons, the thalamic neurons themselves, and see how they affect these behaviors. So first, we silenced SNR projections to the thalamus, and we did this by using this EOPN3, which is a relatively recently developed opsin that specifically inhibits synaptic transmitter release at terminals and is really well suited to block the release the synaptic connection at specific projections. So in this case, we injected this virus expressing this opsin in the SNR and implanted an optic fiber over the ventral thalamus so that we can specifically block synaptic transmission from the SNR to the thalamus. And when we then, again, trained animals on this lever pushing task, we first trained animals to become experts so that they can perform this task as I've shown you before. And then in a subsequent session, when they're already experts, we turn on our LED light or laser light to silence these projections in a subset of trials. And when we do that, we can indeed inhibit the behavior of the mouse as well, where the mouse is not trying to push the joystick, but it just seems like they cannot do it. And I just want to highlight that actually this is the part that the mouse usually uses to do it. And the mouse seems to still want to do the behavior. So we're not just blocking movement of the mouse overall. And indeed, actually, if we wait long enough, the mouse will start pushing the joystick with the other paw that it was not originally trained on. So just to show you kind of how this looks in different animals, on top are the mice were expressing the opsin. When we turn on the inhibition light, a lot of these push trials, so each little circle here represents a lever push. And once they become open circles, that means the push was not successful, so it didn't cross the distance threshold that they needed to achieve to receive their water reward. Whereas in control animals just expressing mCherry, they don't seem to care whether we turned on the laser or not. So to quantify this, in those EOPN3 expressing animals, during the inhibition period, we lose roughly half of the number of successful pushes. And actually, indeed, a lot of the remaining pushes were done by mice somehow finding a different strategy to perform this task. Now, this is what's happening during the lever pushing task. So next, we wanted to see what happens during locomotion. And we thought it's a little bit hard to actually measure locomotion on the head fixed running wheel. So instead, we put a mice on an accelerating rotor rod task where mice are put on a rod and we measure how long it takes for them to fall off the rod at different rotation speeds. And again, these EOPN3 animals fall off the rod significantly earlier than the control animals that express mCherry alone. And surprisingly, this effect was specifically strong at slower rotation speeds, but not as obvious at higher rotation speeds in this accelerating rotor rod task. Now, when we do the same thing, silencing cerebellar projections to the thalamus, we actually did not see any effect on the lever pushing task. And that also kind of makes sense if you remember using the fiber photometry recordings. I showed that there is almost no cerebellar activity during this task that we recorded from. So now silencing these projections also seems to have little effect on the performance of these mice. However, when we then put these mice on the accelerating rotor rod, there is a significant drop in their performance, and these mice fall off significantly sooner than control mice, both at lower as well as higher rotation speeds. So now this is what's happening when we silence the inputs to the thalamus. And of course, the kind of main question is, okay, what happens if we actually silence these neurons in the thalamus that receive both inputs entirely? And for that, we just used a sledgehammer approach to ablate these neurons or kill these neurons by expressing caspase-3. So again, we used our AAV1 transsynaptic viral approach to get a flip, and in this case, flip-dependent free in the thalamus, and then have a tree-dependent caspase-3 expressed in these neurons to ablate them. And when we then now just put these mice on an accelerating rotor rod task, we see a significant drop in performance, both at low speeds as well as high speeds. So now these experiments demonstrate that both the inputs to the thalamus from the basal ganglia as well as the cerebellum, as well as these neurons in the thalamus that receive both inputs, are crucial for these motor learning and motor control behaviors. But one thing that, so everything up until now, what I've shown has been part of this preprint, but one thing that we really wanted to understand more about is what actually do individual neurons in the thalamus do, right? So far, I've just shown you these bulk recordings using photometry, but what we really wanted to see is how do neurons in the thalamus that receive both inputs versus neurons that only receive one of the inputs, or like only receive SNR inputs or only receive cerebellar inputs, whether there's any different activity in those. So we do not have the answer for that yet, but just preliminary, we are using in vivo to photon imaging within the thalamus through a Grün lens now to have a higher resolution of looking at this activity in the thalamus. So this is just some preliminary experiments showing that, yes, we can see the activity of individual neurons in this deep region in the thalamus. Fortunately, we have to implant this Grün lens and kind of be a little bit invasive. But what speaks in our favor is that all the projections that go to these neurons or come from these neurons are kind of mostly coming from a posterior or going to the anterior. So we're not actually damaging any of the projections of these neurons themselves. And what this will allow us to do is combine GCAM plasma imaging with a red reporter virus that could label neurons that receive input from the SNR and the basal ganglia, and then we can compare the activity of these double labeled neurons compared to their surrounding neurons. So this is something that's still ongoing, but just to show you that even with this single neuron resolution, we kind of see the same thing, the similar things that we saw with our bulk fiber photometry recordings, where during this lever pushing task, we see a increased activity on average and pretty much throughout all the neurons that we have recorded from. So, and this activity is particularly stronger during successful pushes compared to non-successful pushes, which reflects this additional signal that happens during the reward period. So these thalamic neurons also encode reward signals, which likely they receive through the SNR inputs. Similar when we put these mice on this cylindrical treadmill, we see a strong activity that's aligned to the motion onset. And yeah, with that, I can just summarize briefly what I've shown you so far, which is that we identified these individual neurons in the motor thalamus that receive synaptic input from the basal ganglia and the cerebellum. These thalamic neurons functionally integrate cerebellar activity, basal ganglia and cerebellar activity. However, with motor learning, the activity in the thalamus adapts differently than what is just happening at its presynaptic inputs in the SNR and the DCN. And silencing either basal ganglia or cerebellar inputs affects these motor behaviors kind of in accordance to how they were active during the behaviors. And silencing the neurons in the thalamus that receive both basal ganglia and cerebellum input also impairs motor learning on the rotor rod task. So with that, I think we can kind of revise or have to revise this traditional model where basal ganglia and cerebellar circuits remain separated in the thalamus and have to think about it a little bit differently, where actually the thalamus has a significant population of neurons that receives inputs from both the basal ganglia as well as the cerebellum, and these neurons functionally integrate the information that comes, or the signals that come from the basal ganglia and cerebellar outputs, and can adapt, and thus probably more likely affect the way that basal ganglia and cerebellar motor signals are transmitted to the motor cortex. So now I would just like to thank the Ding lab, especially my advisor Jun Ding, and people in the lab that helped with the study, particularly Fujin, who helped with the slides electrophysiology, and Charlie Huang, who was an undergrad in the lab and actually was very helpful and is currently a PhD student at Princeton. So with that, I also like to thank my funding sources and thank you all for your attention. And I would love to hear any feedback and ideas that you have.
Thank you so much, Dr. Roth. So we're open to questions. Please raise your hand or unmute yourself if you have any questions. So let me start. So these are very interesting results. So maybe I missed it, but can you please, like, once again, in the DCN responses, did you see any reward? So my understanding was that your SNR responses showed both motor responses and reward responses. Do you have similar in the DCN neurons? And can you give us like an intuition about, like, is it some kind of integration between the reward and movement that is being sent to the cortex?
Interesting. Yeah, that's a great question. So first, looking at the cerebellar signal, we basically don't see any signal here. At early stages, if you quantify the averages, it does look like that there is a significant increase in activity that happens during the reward time. So this would be the 1 to 2 second time interval here. Sorry, this was wrong. And however, at the later stages of learning, where these mice perform more successful pushes and would actually have more reward, this reward signal goes away. And just if you want to compare that to what happens during this whole locomotion task, like this activity is also very much smaller. So that said, I don't think we have a lot of cerebellar reward signals coming to the thalamus here. So the main kind of reward signal that these neurons show would come through the SNR. Or through other sources that we are not probing, right? So one other big input that these neurons get is feedback from the cortex again. So whatever information the cortex has, which will get a lot of things, will also be fed into these neurons again. And yeah, so the question is, okay, do these thalamic neurons mediate some sort of integration between motor and reward signals? I think that is very likely, because these thalamic neurons with learning definitely show a strong response during this reward period. We are right now performing some follow-up experiments where we will either leave out the reward or delay the reward for these tasks to more specifically tease out what signal is actually movement related and what signal is actually reward related. And I think once we have that, we can even better address this question and see how much of this signal that these thalamic neurons show come from the movement or from the reward itself.
Very interesting. Thank you. Austin, please go ahead.
Hey, Richard, that's great talk. Really, really impressive work. My question is about the linear regression fits from the SNR and DCN projections to the thalamus. I mean, it seems to be modeled 90% of the time really, really well, but then there are, kind of spikes in thalamic activity that seem to break that. I'm just wondering what's going on there. Kind of how are you, like, what do you think is changing, perhaps scaling factors between, if the firing is mainly following SNR or DCN, is that neuromodulation? Is that activity in one brain region and the other? Kind of what's going on here?
Yeah, that's a great question. So, yeah, I mean, let me just add like one wrinkle to this, which can explain this, but it makes this even more complicated. So the SNR projections to a thalamus is inhibitory, right? So, and the cerebellar projections to a thalamus are excitatory. And in our case, it always looks like that both of these signals positively contribute to the thalamic activity. You would kind of intuitively expect that the SNR should be inversely correlated to thalamic activity, where the cerebellum should be positively correlated. So one way that we're thinking about it, and which might explain some of these kind of deviations there, especially the larger spikes, is that the thalamic neurons have these characteristic rebound firings following a release of inhibition. So depending on the precise timing on when SNR activity comes to the thalamus, in particular when it ends, there could be kind of our little pet hypothesis that we're trying to do some slides and some more in vivo work to prove or test whether it's true, is that if the release from inhibition from SNR coincides with an activity from the cerebellum, that we then can have some sort of supralinear or nonlinear excitation of the thalamus, which can cause these extremely large thalamic responses, or that if you have subthreshold input from the cerebellum in the beginning, which then coincides with a terminated inhibition from the SNR, you can actually have a meaningful signal in the thalamus that then gets transmitted. Yeah, so right now we're trying to look at the timing of these of the SNR and the cerebellar activity more precisely. It's hard from kind of this bulk calcium imaging experiment to tease that apart. Just preliminary, it does look like that SNR activity is usually a little bit earlier than cerebellar activity, but whether it's like how much release from inhibition do you need to coincide with cerebellar activity, that's something that we're trying both in using slide physiology to model this using optogenetics, or in vivo doing some both neuropixels recordings, but also doing some voltage imaging to look at both subthreshold changes in membrane voltage and have just a much faster readout than using calcium imaging.
Christopher, please.
Hi, Richard. That was a awesome talk. I also really like this what you're just saying about looking at the input, the inhibitory input, and seeing how that interacts with the T types. That sounds really cool. Hopefully one day we'll meet and we can talk about that further in person. But I was actually very curious about your substantia nigra injection. So those viruses are not constrained by any Cree dependence or anything like that, right? So I'm kind of wondering whether there's any structure to the substantia nigra. If you've done kind of mapping experiments, are there different subnuclei within the nigra?
Yeah, that's a great question. So actually, in some of the experiments, so when I do the axonal tracing and also for the silencing experiments using EOPN3, I'm using the VGAT tree mouse. So in that case, I am restricted to inhibitory neurons, but I'm still targeting kind of as much of the SNR as possible. So basically, in this case, both for the SNR as well as for the cerebellar nuclei, we took the approach kind of we want to cover as much of them as possible to get kind of an upper limit of the integration that could happen in the thalamus. And so with that, like right now, I don't have any information about kind of subnuclei of both the cerebellum or the SNR. But like you said, the SNR has a lot of different subdivisions that are known to project to different places throughout the brain. They all seem to have collaterals that go to the thalamus, as far as I understand. Like basically, there are some that are preferentially going more to the hindbrain, some that are preferentially more going to striatum and anterior parts of the brain. But looking at these studies that have done more single cell tracing of the axonal arbors, most of them have collaterals that go to the thalamus. But yeah, I think that would be also something interesting to follow up on later, is like do very precise injections within the SNR to see whether it's like more the medial or the lateral SNR that has stronger integration or interactions with the cerebellar nuclei in the thalamus.
Thank you. That was great.
Alan, please go ahead.
Yes, super interesting talk. Thank you. I'm wondering, given that the extent of this pathway where both of these inputs come together is relatively small compared to the parallel inputs, functionally considering these, two parallel and one integrating pathway, what do you think is the relative function potentially? Can you speculate on that?
Yeah, that's a great question. So, I mean, I think it's not necessarily that much minor to the parallel circuits, right? So we kind of the best quantification that we have is from our patch recordings, where roughly 30% of the neurons that we reported from receive both of these inputs. And let's say, I mean, I think our cerebellar inputs are probably a little bit undersampled here because most of the neurons that we reported from are more medial and not in a VL where we would expect more cerebellar inputs. So maybe just ballpark, there would be a third, and a third of the population. So sure, there's 2/3 that are kind of parallel and 1/3 that's integrated. And so it's possible, we're starting to look a little bit at this, whether these different populations, kind of like, your question is like, what is the functional role that these different populations could have? And we honestly do not have the answer to this right now. But one thing that did stick out a little bit is when we look at the projections of those to the cortex. So we have not carefully analyzed that yet, that's why I'm not showing that, but it does look like that the neurons with dual input do project to slightly different places than the ones that either have SNR only or cerebellar only, particularly where these neurons that have both inputs seem to go more to the primary motor cortex, whereas the other ones go more to secondary motor cortex. So it's possible that there is kind of a little bit of a shorter loop where they can affect primary motor cortex outputs more directly than these neurons that are part of these more parallel circuits. And maybe these parallel circuits could go up to higher order cortical regions where then higher order integration could happen. Whereas when the integration already happens in the thalamus, that can be fed into the primary motor outputs with a couple synapses fewer. But yeah, in order to really understand this, we really want to record from the activity of these three populations separately so that we can see, for example, do these thalamic neurons in the cerebellum really do not have any reward information? So maybe like one important function of these neurons in the thalamus would be to bring reward-related information from the basal ganglia system into kind of the more movement adaptation or sensory adaptation information that comes through the cerebellum. But yeah, I mean, that's all speculation right now. And we're trying to record from these populations simultaneously so that we get a better sense of exactly what their functional differences might be.
Okay, great. Thank you.
Great, thank you. Are there any other questions? Okay, thank you so much. It was great having you.
Thank you. Yeah, thanks for all the questions.
Great.
It's a joy to present to Hopkins again.
Oh, sorry. Rick, do you have a question?
Just wanted to say it's good to see him.
Good to see you too.
Great talk.
Great.
Thanks.
Have a great rest of your Tuesday. Goodbye.
Thanks. You too. Bye.